52 million years ago, something was already flying through the night sky. The fossil was discovered in Wyoming, preserved in remarkable detail. It had wings, it could hunt in the air, it looked unmistakably like a bat, and that is the problem. The oldest bat ever found was not halfway to flight. It was not experimenting with flight. It was already a bat. Whatever came before it is missing. The creature that first stretched its fingers into wings, left the trees, and entered the night has never been found. The deeper scientists looked, the stranger the story became. Today, there are more than 1,400 species of bats, making up more than one in every 5 mammal species on Earth. They are the only mammals capable of true powered flight. They navigate in complete darkness using sound. Some live decades longer than animals their size should. Some carry viruses that barely affect them, but can devastate other species. And one group evolved a blood thinning compound so effective that scientists transformed it into a potential treatment for human stroke patients. By every normal biological rule, bats should be impossible. Their flight is unusual, their immune systems are unusual, even the way they age appears unusual. For more than 2 centuries, scientists have tried to understand how a small nocturnal mammal became one of the most successful and bizarre animal groups on the planet. Tonight, we're exploring the mystery of how bats became the strangest mammals on Earth. But before we do, let me know in the comments, are you listening to this to fall asleep, or you're here only for the knowledge? Settle in and let's uncover the missing chapter that may explain how mammals first conquered the night sky. A scar lay across the history of life, long before the first bat entered the sky. Around 66 million years ago, an asteroid struck near what is now the Yucatan Peninsula, bringing the age of the non-avian dinosaurs to an abrupt close. Forests burned, food webs collapsed, and roughly 3/4 of all species disappeared. Across land, sea, and air, familiar rulers vanished within a geological moment. The survivors were rarely the largest or strongest creatures. Many belonged to lineages that had spent more than 100 million years existing at the edges of ecosystems. Small mammals moved through undergrowth, sheltered in burrows, and searched for food during the hours when larger predators were less active. Their world was one of insects, seeds, roots, and caution. Generation after generation had been shaped by survival in the background. When the catastrophe passed, the planet entered the Paleocene epoch. The forests returned. Warm temperatures spread across much of the globe, and moisture supported dense vegetation from lowlands to river valleys. Ecological space opened in every direction. Entire ways of life had vanished, leaving opportunities that had not existed for tens of millions of years. Birds inherited the daylight skies. Their wings already carried them across continents, shorelines, and forest canopies. Daytime flight belonged to them. Yet the hours after sunset offered a different landscape. Darkness concealed immense swarms of insects. Night air drifted between branches largely unclaimed. No highly specialized aerial hunter dominated those conditions. Within tropical and subtropical forests, life unfolded among trunks, vines, and layered canopies. Countless insects emerged after dusk, filling the air with movement. Small mammals climbed, leaped, and searched among branches. Somewhere within this setting, a lineage began moving toward an evolutionary destination unlike any reached before. The creature responsible remains absent from the fossil record. No skeleton has revealed its proportions. No preserved bones show the gradual reshaping of fingers into flight surfaces. Its appearance, behavior, and exact habitat remain unknown. The evidence available today comes from later descendants and from the genetic signatures carried within living bats. Molecular studies suggest that the ancestors of bats diverged from other mammalian groups sometime between roughly 64 and 80 million years ago. That estimate places their origin near the boundary between a world dominated by dinosaurs and a world newly open to mammalian experimentation. For millions of years afterward, the record falls into darkness. Stone preserves no clear account of what happened there. What can be said with confidence is that the transformation must have been extraordinary. Across more than 300 million years of mammalian ancestry, no lineage had developed true powered flight. Many animals climbed, some glided, others leaped impressive distances between branches. None crossed the threshold that bats eventually crossed. The forests of the Paleocene provided opportunity, food, shelter, and time. Evolution worked within those conditions, reshaping an animal that has never been found into one that would eventually appear fully formed in the rocks. By the time the story becomes visible again, the outcome is already waiting in the air, carrying with it one of the most elusive origins in the history of mammals. In a slab of pale stone from Wyoming, a small skeleton rested for more than 50 million years before anyone realized how disruptive it would become. When researchers described the specimen in 2003, they were looking at an animal preserved with extraordinary clarity. Delicate bones remained in place. Traces of soft tissue survived. The outline was unmistakable. Its name was Onychonectarus finii. At roughly 52.5 million years old, it became the oldest bat fossil known to science. Age alone would have made it important. Its true significance emerged from what its body revealed. The fossil possessed elongated fingers supporting broad wing membranes. Its shoulders, forelimbs, and chest carried the hallmarks of powered flight. Nothing about the skeleton suggested a creature experimenting with the air. Nothing indicated a tentative step toward becoming a bat. The architecture was already present. The animal could fly. That discovery created an unusual problem. In many evolutionary stories, the path remains visible. Fossils preserve intermediate stages, allowing researchers to follow gradual changes through time. Bird evolution offers one of the clearest examples. Specimens such as Archaeopteryx and numerous feathered dinosaurs document a sequence extending across millions of years. Bone by bone, feature by feature, the transition can be traced. The bat record tells a different story. When the curtain rises, the main character is already on stage. The creatures expected to stand between a climbing mammal and a flying bat have not been found. No confirmed fossil shows partially elongated fingers. No preserved skeleton captures a wing membrane midway through its development. The earliest chapter remains absent. Geology provides a practical explanation. Small animals leave delicate remains. Thin bones break easily, scatter readily, and survive burial less often than the heavier skeletons of larger creatures. Conditions matter as much as anatomy. Dense tropical forests, where many researchers suspect early bat evolution occurred, are poor environments for fossil preservation. Moist soils, active roots, scavengers, and rapid decomposition erase traces before stone can record them. Time may have contributed another complication. Molecular studies comparing mutation patterns across living bat lineages suggest their ancestry extends much deeper than the oldest known fossil. Many estimates place the origin of bats between 64 and 80 million years ago. If those calculations are correct, a substantial stretch of bat history exists beyond the reach of known fossils. Some paleontologists propose that the transformation unfolded with surprising speed. A span of 5 to 10 million years may sound immense from a human perspective, yet within geological history, it can pass almost as a brief episode. If transitional populations remained small and existed for a comparatively short interval, opportunities for preservation would have been limited. For that reason, researchers often describe early bat history as a ghost lineage. The term does not refer to anything supernatural. It describes an evolutionary branch inferred from genetics and later fossils, despite the absence of direct physical evidence. The lineage is visible through its consequences, while much of its actual record remains missing. Onychonycteris finii sits at the edge of that absence, illuminating a history it cannot fully reveal. Its bones confirm that bats had already mastered flight by the time they entered the fossil record, leaving one of vertebrate paleontology's most enduring gaps suspended between stone and inference. Look closely at a batwing, and the strangeness begins to resolve into familiarity. The shape seems foreign at first, a dark membrane stretched into motion, yet its framework comes from an inheritance shared with every human hand. The scientific name for bats is Chiroptera, from Greek words meaning hand wing, and the name is exact in a way that feels almost anatomical before it feels poetic. Inside that wing are the same basic bones found in your own arm and hand. There is a forearm, a wrist, a thumb, and four fingers. Evolution did not create a new limb from nothing. It took an old mammalian structure and changed its proportions until the hand became large enough to hold the air. In many bats, the fingers are the true wing spars. The greater horseshoe bat offers a striking example. Its third finger can extend longer than the animal's entire body, with the fourth and fifth fingers nearly as extended. These digits no longer serve mainly for grasping. They have become flexible supports, spreading the flight surface with a precision that rigid feathers could never provide. The thumb, smaller and clawed, remains free from the membrane, still useful for climbing, gripping bark, and moving across roost surfaces. The membrane itself is called the patagium. It is often described as skin, though that word makes it sound far simpler than it is. The patagium contains blood vessels, nerves, connective tissues, elastic fibers, and sheets of specialized muscle running through the membrane. Among these are the plagiopatagiales, muscle bands that allow the bat to alter wing shape during each wing beat. That ability changes everything. A bird wing is built around feathers and a stiffened forelimb. A bat wing behaves more like a living sail under muscular control. As air flows across it, the bat can adjust tension, curvature, and surface angle in fractions of a moment. The wing does not merely flap, it reshapes itself while moving, tightening, flexing, and relaxing as the animal turns, climbs, breaks, or reaches for prey. The deeper story begins before birth, inside the developing embryo. Mammals share many of the same developmental instructions for building limbs, and bats did not abandon that toolkit. They changed how strongly certain instructions were expressed. One important gene, called paired related homeobox 1, helps guide forelimb growth in mammals. In bat embryos, this gene shows far greater activity in the growing forelimb than it does in mouse embryos at the same stage. Experiments have shown how powerful that switch can be. When researchers increased paired related homeobox 1 activity in developing mouse limbs, the forelimb bones grew measurably longer. The machinery for elongation was already present in mammals. In bats, evolution turned that machinery upward while also reducing signals that normally limit digit growth. The change required both release and acceleration. The thumb escaped this transformation because it follows a different developmental path. It stayed compact, clawed, and useful, while the other digits lengthened into a framework for flight. The result was one of the most radical alterations of a vertebrate limb ever produced, a hand remade into a wing without ever ceasing to be a hand. Among the many paths bats have taken through evolutionary history, one leads to a feeding strategy so unusual that it seems almost disconnected from the rest of mammalian life. Deep within parts of Latin America, three bat species survive on a diet composed entirely of blood. The common vampire bat, the white-winged vampire bat, and the hairy-legged vampire bat occupy a niche that no other mammal has mastered with such precision. Their approach begins with anatomy refined over millions of years. The upper front teeth and canine teeth form cutting instruments of extraordinary sharpness. When a vampire bat lands on a sleeping animal, it does not tear flesh or leave a ragged wound. A small incision is opened with remarkable accuracy, often measuring only a few millimeters across and deep enough to reach vessels near the surface. Many hosts remain unaware that feeding has begun. Finding the right location requires another specialized adaptation. Around the face are sensory structures capable of detecting subtle differences in heat. Warm blood flowing close to the skin creates patterns that guide the bat toward suitable feeding sites. Similar heat-sensing systems evolved independently in pit vipers, a reminder that distant branches of life sometimes arrive at comparable solutions when confronted by similar challenges. The feeding itself is frequently misunderstood. Vampire bats do not draw blood through suction. After making the incision, they use the tongue to lap liquid from the wound. This method depends on chemistry as much as anatomy, because blood naturally begins clotting almost immediately after tissue damage occurs. Without a way to prevent clot formation, feeding would become difficult and inefficient. Inside the saliva lies the key. Vampire bats produce a specialized anticoagulant protein known as draculin. Once introduced into the wound, it interferes with the clotting process and keeps blood flowing long enough for the animal to feed. What appears to be a simple meal is supported by a biochemical system refined through countless generations of natural selection. That same molecule eventually attracted the attention of medical researchers. During the 1990s, scientists isolated and studied draculin, recognizing that its ability to influence blood flow might have applications far beyond bat biology. Those investigations contributed to the development of a related therapeutic compound called desmoteplase, designed to dissolve blood clots in patients experiencing ischemic stroke. Stroke treatment is governed by time. Existing clot-dissolving therapies are most effective within a narrow window after symptoms begin. Clinical studies have suggested that desmoteplase may remain useful over a longer interval, potentially allowing treatment for patients who arrive too late for conventional options. Research has progressed through advanced stages of clinical testing as investigators continue evaluating its effectiveness and safety. The story creates an unexpected connection between cave roosts and hospital wards. A feeding adaptation shaped by roughly 30 million years of evolutionary pressure became a source of insight for modern medicine. The vampire bat evolved draculin to solve a practical problem encountered during nightly feeding. Human researchers encountered the same chemistry and discovered a tool that may help preserve blood flow within the injured brain. In that way, a creature long associated with fear and folklore contributes something entirely different, a biological solution transformed into clinical pharmacology. Biologists have long relied on a pattern that appears across almost every branch of the mammalian family tree. Large animals tend to live long lives. Small animals tend to live short ones. The rule is so dependable that body size alone often provides a reasonable estimate of lifespan. Tiny mammals burn energy rapidly, accumulate cellular damage quickly, and usually disappear after only a few years. A bat should be no exception. Most bats weigh only a few grams. Their lives are powered by flight, among the most demanding activities any vertebrate can perform. Every night, muscles consume tremendous amounts of energy. Hearts race, breathing accelerates. From a physiological perspective, the body seems built for rapid wear. According to standard metabolic models, a mammal weighing around 8 grams should survive perhaps 3 to 4 years. Other small mammals support that prediction. A shrew may live roughly 18 months. A mouse often survives about 2 years. Generation after generation, the pattern repeats with striking consistency. Then bats enter the dataset and refuse to cooperate. The little brown bat has produced verified records of wild individuals living beyond 34 years. Brandt's bat, a species scarcely larger than a human thumb, has yielded one of the most astonishing records ever documented. A banded individual, later recovered in Russia, was confirmed at 41 years of age. For an animal of its size, such longevity is extraordinary. It represents roughly 10 times the lifespan expected under conventional biological assumptions. Scientists initially searched for a single explanation. What emerged instead was a collection of interconnected adaptations, each contributing to a slower pace of aging. One important clue comes from telomeres, protective structures located at the ends of chromosomes. In many organisms, telomeres gradually shorten as cells divide. Their erosion is closely associated with aging and declining tissue function. Several long-lived bat species show unusually stable telomeres, allowing cells to continue functioning effectively over much longer periods. Researchers then turned their attention to mitochondria, the structures responsible for producing cellular energy. Energy production inevitably creates reactive oxygen species, molecules capable of damaging proteins, membranes, and genetic material. Across decades, that damage accumulates. Studies of long-lived bats have identified genetic variants associated with greater mitochondrial efficiency, reducing the production of these harmful byproducts and limiting the rate at which cellular injuries build up. Another piece of the puzzle involves the mechanistic target of rapamycin pathway, a molecular signaling system linked to growth and aging across a wide range of organisms. During torpor, when many bats dramatically lower their metabolism, activity within this pathway declines. Resources are redirected toward maintenance and repair, helping preserve tissues through repeated cycles of dormancy and recovery. Protection extends even deeper. Genes responsible for repairing damaged deoxyribonucleic acid show elevated activity in several bat species. Everyday molecular injuries that accumulate in most animals are corrected more efficiently, preserving the integrity of the genome over time. None of these mechanisms operates in isolation. Together, they create a biological strategy that slows the progression of aging itself. Damage accumulates more gradually. Repair systems remain active. Cellular function is preserved far longer than expected. For scientists studying human longevity, bats have become far more than an unusual curiosity. They provide living evidence that mammalian aging is not fixed to a single timetable. Within a body small enough to rest in the palm of a hand, evolution assembled a system capable of stretching life across decades, revealing possibilities that biology once seemed to place beyond reach. A virus entering a mammalian body usually triggers a familiar sequence of events. Cells detect the invader. Alarm signals spread through surrounding tissue. Inflammation rises. Immune defenses mobilize. The process is designed to contain infection quickly, even at the cost of collateral damage to the host itself. For most mammals, that strategy works well enough. Bats live by different rules. Across the world, researchers have identified bats as natural reservoir hosts for an extraordinary collection of viruses capable of infecting humans and other mammals. The list includes ancestral relatives of severe acute respiratory syndrome, coronavirus 1, and severe acute respiratory syndrome, coronavirus 2, along with Ebola virus, Marburg virus, Nipah virus, Hendra virus, Middle East respiratory syndrome, coronavirus, and many additional viral lineages discovered through genetic surveys. The diversity is so extensive that new viral families continue to be identified as sampling expands into previously unstudied regions. What makes this remarkable is not the presence of the viruses themselves, it is the condition of the host carrying them. In many vertebrates, these pathogens cause severe disease. Organs become damaged, tissues become inflamed. The immune response can become so intense that it contributes more to the illness than the virus itself. During severe infections, the body's own defenses may create widespread injury while attempting to eliminate the invader. Bats frequently avoid that outcome. The explanation begins with the innate immune system, the ancient first layer of defense shared across vertebrate evolution. This system reacts rapidly to infection and relies on molecular sensors capable of detecting foreign genetic material. One important component is known as the stimulator of interferon genes pathway. In many mammals, activation of this pathway triggers a powerful inflammatory cascade intended to destroy infected cells and prevent viral spread. In bats, that signaling system has been modified. The relevant gene is truncated, reducing the intensity of the inflammatory response. Detection still occurs. Antiviral activity still follows. The destructive escalation that often accompanies infection is greatly reduced. At the same time, bats maintain another unusual adaptation. Their interferon defenses remain active even when infection is absent. Rather than waiting for a pathogen to arrive before mobilizing protection, parts of the antiviral system operate continuously at a low baseline level. Viral replication is constrained early, often before it reaches levels that would provoke a damaging inflammatory reaction. The origin of this arrangement appears to trace back to flight itself. Sustained, powered flight places extraordinary demands on the body. During intense activity, core temperatures can rise by 1 to 3 degrees Celsius or 2 to 5 degrees Fahrenheit. Metabolism accelerates dramatically. Cellular stress increases. Deoxyribonucleic acid damage occurs more frequently than it does in less active mammals. An immune system responding aggressively to every instance of cellular damage would create constant inflammation. Over evolutionary time, bats appear to have solved that problem by dampening inflammatory pathways while strengthening antiviral defenses. The result is a body capable of tolerating conditions that would overwhelm many other mammals. This adaptation was never aimed at becoming a viral reservoir. Natural selection favored animals capable of sustaining flight without harming themselves through chronic immune activation. Viral tolerance emerged alongside that solution. The consequence extends far beyond the bat colony. By reshaping the relationship between infection and inflammation, evolution produced one of the most sophisticated immune systems known in mammals, a system that altered the course of disease ecology across the planet and continues to influence the history of our own species. Sound leaves the bat as a brief pulse and returns a fraction of a moment later, carrying information. Within that interval, an entire landscape has been measured. Distance, direction, texture, movement, and shape arrive together, encoded within echoes that would pass completely unnoticed through the human world. The calls themselves occupy frequencies far beyond ordinary hearing. Depending on the species, echolocation signals may range from roughly 20 kilohertz to 200 kilohertz. As the sound spreads outward, it strikes leaves, insects, branches, cave walls, and open air. Each object reflects a slightly different signature. The returning echoes become raw material for a sensory system unlike any possessed by humans. The challenge is immense. The bat is moving. The insect is moving. Air currents alter trajectories. Obstacles appear from every direction. Despite this complexity, processing occurs within milliseconds. By the time one pulse has returned, another is often already on its way. Laboratory experiments reveal the precision of the system. Echolocating bats have successfully detected and avoided wires, measuring only 0.08 millimeters in diameter. Such a wire is thinner than a human hair and difficult to see even under bright illumination, yet the bat identifies it through reflected sound alone while remaining in motion. Accomplishing this feat required profound changes within the brain. In several species, regions devoted to auditory processing occupy an unusually large proportion of total brain volume. Neural resources that many mammals devote primarily to vision are redirected toward the interpretation of acoustic information. Evolution did not merely sharpen hearing, it reorganized perception around it. Within a structure known as the inferior colliculus, individual neurons are tuned to specific echo delays. Some respond only when an echo returns after a particular interval. Others respond to slightly different delays. Together, they create a spatial arrangement of neural activity corresponding to distance. The brain effectively transforms time into space, constructing a map from fractions of a second. This process produces something difficult to compare with ordinary human experience. The bat is not listening in the way humans listen. It is continuously generating a three-dimensional representation of its surroundings using reflected sound. Every pulse updates that model. Every returning echo refines it. One additional challenge remains. Echolocation calls can be extraordinarily loud at their source. Without protection, a bat could overwhelm its own hearing every time it emitted a pulse. Evolution solved this problem through exquisite timing. Just before a call is produced, a small muscle within the middle ear contracts. This action temporarily reduces hearing sensitivity during the outgoing signal. The muscle then relaxes almost immediately, allowing the returning echo to be detected with exceptional clarity. The precision involved is measured in microseconds. These adjustments occur automatically hundreds of times during an active hunting period. No conscious effort is required. The system functions as seamlessly as breathing. Engineers have spent decades studying this sensory architecture. Research, funded by the United States Navy, examined aspects of bat hearing in hopes of improving sonar technology. The cochlea, auditory pathways, and signal processing strategies found in bats continue to inspire new approaches to underwater detection and acoustic imaging. Even after years of investigation, the biological solution remains astonishing. A mammal entered the darkness, embraced sound as its primary guide, and reshaped its brain around that decision. The result is a sensory system capable of extracting detail from echoes with a precision that human technology is still striving to match. Classification systems often appear stable until a new piece of evidence arrives and rearranges the entire picture. For much of the 20th century, bats seemed neatly divided into two groups. One contained the smaller species that navigated through darkness using sophisticated echolocation. The other contained the large fruit bats, often called flying foxes, animals whose enormous eyes suggested a different relationship with the night. The distinction appeared straightforward. Small bats produced laryngeal sonar calls and relied heavily on acoustic navigation. Flying foxes depended primarily on vision and, in most cases, lacked the elaborate echolocation systems found in their smaller relatives. Taxonomists therefore separated bats into two suborders. Microchiroptera contained the echolocators. Megachiroptera contained the fruit bats. The arrangement implied that laryngeal echolocation evolved once and was inherited by all descendants within the echolocating branch. As genetic technology matured during the late 1990s, researchers gained access to a different source of evidence. Bones, teeth, and anatomy remained important, yet genomes preserved a deeper historical record. When scientists compared genetic relationships among bat families, the results challenged decades of accepted classification. Several echolocating groups turned out to share a more recent common ancestry with fruit bats than with other echolocating species. The old division no longer matched the evolutionary tree emerging from molecular evidence. Branches that appeared separate through anatomy alone were intertwined through genetics. This discovery carried an extraordinary implication. If the genomic relationships were correct, then laryngeal echolocation could not have arisen only once and passed unchanged through a single lineage. The evidence suggested that complex sonar systems emerged independently in more than one branch of bat evolution. Evolution had assembled comparable biological machinery multiple times. Such convergence is known elsewhere in nature. Wings appeared independently among insects, pterosaurs, birds, and bats. Camera-like eyes evolved in both vertebrates and cephalopod mollusks. Echolocation, however, demands an especially intricate integration of structures. Sound production, auditory processing, neural timing circuits, specialized hearing organs, and behavioral coordination must all function together. Reconstructing that entire system more than once elevates convergence to an exceptional level. Additional support arrived from studies examining hearing-related genes. Among the most discussed was a gene known as prestin, which influences the behavior of outer hair cells within the cochlea. Researchers discovered parallel genetic modifications in echolocating bats and dolphins, two mammalian lineages separated by roughly 90 million years of evolutionary history. The similarity was striking enough that some investigators initially suspected contamination or analytical error. Independent laboratories repeated the work and reached the same conclusion. Distinct groups of mammals facing comparable acoustic challenges had altered some of the same genetic pathways in comparable directions. Patterns such as these reveal something important about natural selection. Evolution does not operate with foresight, yet physical reality imposes constraints. Sound behaves according to consistent laws. Biological tissues obey the same chemistry. Under certain circumstances, different lineages encounter similar problems and discover similar solutions through separate evolutionary journeys. Within the bat lineage, sonar appears less like an improbable accident and more like a destination, repeatedly favored by selection. The outcome emerged through different branches of ancestry, shaped by the same demands of navigating and hunting in darkness. The echoes crossing the night carry evidence of that remarkable convergence, preserved not only in behavior but also within the genes themselves. Evolution usually leaves the impression of patience. Lineages branch, bodies adjust, habits shift, and the record gathers those changes across immense spans of time. Bats challenge that rhythm. When they enter the visible record around 52 million years ago, they are already more than a single experiment in flight. They are already separating into different ways of being a bat. Within only a few million years, their diversity becomes difficult to ignore. By roughly 45 million years ago, fossil evidence and molecular comparisons point toward lineages connected to the ancestors of modern insect hunters, fruit eaters, nectar feeders, carnivores, and blood feeding specialists. A mammalian order that had only recently become visible was already spreading into ecological roles, normally associated with far longer evolutionary histories. This kind of expansion is called adaptive radiation. It occurs when a lineage enters an open ecological zone and diversifies rapidly to occupy many available niches. In bats, the process moved with unusual speed. The comparison with primates is revealing. Lemurs, monkeys, apes, and humans represent an order whose diversity accumulated across more than 65 million years. Bats reached a comparable breadth of ecological strategy in a fraction of that span. The force behind that expansion came from a rare combination of abilities. Powered flight gave bats access to the air. Echolocation opened the night with precision. Together, these traits allowed mammals to enter spaces that had remained largely unavailable to their relatives. Insects flying after dusk, fruit hanging beyond the reach of ground animals, nectar blooming under night skies, cave systems, tree hollows, and narrow aerial corridors all became part of the bat world. Their body plan also proved unusually adaptable. A membrane wing could maneuver through cluttered forests, skim over water, hover near flowers, or sweep through open air. Teeth, tongues, skulls, ears, and digestive systems shifted as lineages specialized. One ancestral framework generated many feeding styles without abandoning the core design that made bats bats. Modern genomics has shown that this speed was not confined to outward form. In 2021, a whole genome sequencing study of 37 bat species produced an evolutionary tree that overturned several older assumptions about bat family relationships. Some branches had separated so early and so quickly that even detailed genetic data required researchers to revise long accepted groupings. A major study published in 2020 examined 40 bat genomes and identified more than 1,000 genomic regions showing accelerated positive selection compared with other mammals. These regions involved immunity, metabolism, sensory biology, development, and deoxyribonucleic acid repair. The same rapidity, visible in wings, skulls, and feeding behavior, was also written into the molecular record. Biologists sometimes describe such bursts as quantum evolution, rapid large-scale change driven by entry into an underused adaptive zone. The phrase does not imply magic. It names a tempo faster than ordinary gradual expectation, where opportunity and flexible biology meet. Today, 21 bat families are recognized, and their relationships continue to be refined. The order did not spend tens of millions of years cautiously becoming diverse. It expanded early, quickly, and with unusual force, leaving behind a living radiation that still fills the night with forms descended from that first burst of becoming. At dusk in central Texas, the entrance to Bracken Cave begins to change shape. What first appears to be a dark opening in limestone becomes the source of a moving column, a living current rising into the evening sky. Minute by minute, the stream thickens until individual animals become impossible to distinguish. The eye stops counting. Mathematics must take over. Each spring, Approximately 20 million Mexican free-tailed bats gather here to give birth and raise their young. No other known concentration of mammals reaches this scale. The colony transforms a single cave in Comal County into one of the most densely populated biological spaces on Earth, a seasonal city measured not in streets or buildings, but in wings and heartbeats. The evening emergence unfolds over several hours. Bats pour from the cave in a spiraling ribbon that can stretch across the landscape and climb high into the atmosphere. The biological cloud is so large that weather radar routinely detects it. Meteorologists in the San Antonio region have occasionally mistaken these signals for approaching storm systems before recognizing their true source. A radar screen designed to track weather becomes, for a moment, a map of animal movement. Once airborne, the colony spreads across the surrounding countryside in search of food. The numbers remain difficult to grasp. Researchers estimate that the bats consume approximately 140 tons of insects each night. That figure represents countless moths, beetles, leafhoppers, and agricultural pests removed from the ecosystem before they can reproduce or damage crops. Across an entire summer season, the cumulative effect becomes enormous, providing ecological services worth millions of dollars to nearby farming regions. Inside the cave, another numerical challenge unfolds. Each mother gives birth to a single pup and leaves it behind while foraging. Upon returning, she must locate that one offspring among millions of nearly identical young. The problem resembles searching for a single voice within a crowded stadium many times larger than any human gathering. Field studies have shown that mothers solve this task through several overlapping systems. They return to familiar roost locations with impressive spatial accuracy. Each pup develops a distinctive vocal signature that its mother learns and recognizes. At close range, scent provides additional confirmation. Together, these cues create a reliable identification process. Despite the overwhelming density of the colony, reunification success exceeds 80% during returns from nightly feeding trips. The colony also generates its own environmental conditions. Millions of small mammalian bodies release heat continuously, creating temperatures favorable for pup growth and development. No architect designed this incubator. It emerges automatically from the combined metabolism of the animals themselves. Individual bats contribute a tiny amount. Collectively, they reshape the climate of an entire cave chamber. Year after year, guano accumulates across the cave floor. Over thousands of years, these deposits have supported a separate community of organisms, including specialized beetles, mites, moths, fungi, and microorganisms adapted to conditions found nowhere else. Their existence depends entirely on the bats above them. Evidence suggests that bat colonies have occupied Bracken Cave for roughly 10,000 years. Through changing climates, shifting landscapes, and the rise of human civilizations, the colony has persisted as a biological foundation supporting an entire subterranean world built from abundance, memory, and continuity. A sheet of living membrane stretches between elongated fingers, yet calling it skin leaves out most of the story. Calling it fabric misses the point entirely. The structure that carries a bat through the air belongs to a category of its own, one that engineers continue to examine with a mixture of admiration and frustration because no manufactured material fully reproduces its capabilities. Viewed under magnification, the patagium reveals an architecture of remarkable complexity. It consists of an outer epidermis, a middle layer of connective tissue, and an inner epidermis that mirrors the outer surface. Between these layers lies a network of elastin and collagen fibers arranged in a precise geometric pattern. The arrangement is neither random nor uniform. Every strand contributes to how the membrane bends, stretches, and recovers during flight. Its most important property is anisotropy, a term used when a material behaves differently depending on the direction of force applied to it. Pull the membrane one way and it stretches readily. Pull it another way and resistance increases. This directional flexibility gives the wing control instead of instability. Air pressure shifts continuously during flight, and the membrane responds with corresponding adjustments, maintaining aerodynamic performance across a wide range of conditions. Threaded through this structure are the plagiopatagiales, bundles of smooth muscle embedded directly within the wing membrane. These muscles allow the animal to alter tension and curvature while airborne. Every wing beat becomes an opportunity for refinement. The shape of the flight surface changes moment by moment as the bat reacts to moving prey, shifting air currents, vegetation, cave walls, and countless other variables encountered during nocturnal travel. Engineers describe this capability as continuous compliant morphing. Modern aircraft rely largely on rigid surfaces and hinged control structures. A bat operates according to a different principle. Its wing is never truly fixed. Shape adjustment occurs continuously across the entire membrane, creating a level of aerodynamic responsiveness that current aerospace technology has not fully matched. Wind tunnel experiments conducted at Brown University and Lund University revealed another layer of sophistication. High-speed imaging showed bats generating leading-edge vortices, rotating columns of air that increase lift. Conventional aircraft often encounter similar vortices as an operational challenge. The bat produces them predictably and exploits them as part of normal flight. Airflow that would trouble many engineered systems becomes a useful resource. Measurements taken during low-speed flight have produced equally striking results. Bat wings achieve lift coefficients exceeding those of conventional airfoil designs operating under comparable conditions. Near obstacles, during landing maneuvers, and in dense environments, the advantages become particularly apparent. The wing remains effective where many human flight systems lose efficiency. Durability adds another dimension. The membrane resists tearing at levels comparable to surgical materials. When damage occurs, failure tends to remain localized instead of spreading across the entire surface. Materials scientists describe this behavior as graceful degradation. Function is preserved even after injury. Repair follows with similar elegance. Dense networks of blood vessels support rapid tissue regeneration, allowing punctures and tears to close within days. Research teams studying soft robotics, prosthetic design, and flexible electronics continue searching for ways to replicate both the structural weave and the regenerative architecture. By measurable engineering standards, the bat wing remains the most sophisticated biological flight surface ever analyzed, refined through more than 50 million years of evolutionary testing and still outperforming many of humanity's most advanced designs. Survival can sometimes depend on a single successful night. For the common vampire bat, that reality shapes nearly every aspect of life. Blood is a nutritionally specialized food, rich in some resources yet limited in others, and the animal's metabolism consumes its available energy with relentless speed. An individual that fails to feed for roughly 60 hours approaches a physiological threshold from which recovery becomes unlikely. Few mammals live so close to the edge of starvation while appearing outwardly healthy. Such vulnerability created unusual evolutionary pressures. A missed feeding opportunity is never merely an inconvenience. It can become a matter of life and death. Within vampire bat colonies, natural selection responded by fostering a system of cooperation that stands among the most sophisticated known outside humans and a handful of highly social mammals. The foundation of this discovery emerged through the work of researcher Gerald Wilkinson at La Selva in Costa Rica. Years of observation revealed a pattern that challenged traditional assumptions about animal behavior. Well-fed bats frequently regurgitated portions of their blood meals and offered them to roost mates that had returned hungry. The donated food carried real cost. Blood acquired during a night's foraging represented precious energy. Giving part of it away reduced the donor's own reserves. Kinship explained some of the sharing, though not all of it. Many donations occurred between unrelated individuals. A deeper pattern gradually emerged. Bats formed enduring social relationships, and those relationships influenced future decisions. Individuals that received assistance often returned the favor during later periods of need. Cooperative histories accumulated across months and years, creating a network of remembered exchanges. Researchers describe this behavior as reciprocal altruism, the conditional transfer of a costly benefit with the expectation that assistance may later flow in the opposite direction. In the animal kingdom, convincing examples remain uncommon. Among vampire bats, the evidence became difficult to ignore. Individuals that repeatedly accepted aid without contributing in return gradually lost access to the generosity of their colony companions. Reputation carried consequences. Additional studies expanded the picture. Tracking data and acoustic monitoring demonstrated that the strongest food-sharing partners often traveled to similar feeding grounds. Social bonds influenced movement patterns beyond the roost itself. Information about profitable hunting opportunities may pass through these relationships, strengthening cooperative connections that already provide nutritional insurance. Memory plays a central role in maintaining the system. A vampire bat can distinguish among dozens of colony members and maintain separate social histories with each. Relationships differ in strength, reliability, and frequency of interaction. Such complexity exists within a brain scarcely larger than a grape. Grooming contributes another layer. Extended sessions of fur licking remove parasites and debris, yet they also reinforce social ties. Researchers found that grooming frequency predicts future food sharing more accurately than genetic relatedness. Trust appears to be built through repeated contact long before an emergency arises. When long-term companions are separated experimentally and reunited weeks later, vocalizations and behavior shift immediately. Recognition is unmistakable. Familiar partners receive attention that strangers do not. From these interactions emerges something resembling a social economy, sustained through memory, cooperation, reputation, and mutual dependence. Hanging from cave ceilings in darkness, vampire bats built a system whose underlying logic echoes principles found throughout human societies, proving that complex cooperation can arise in forms far removed from our own. Across open country at night, distance changes the meaning of navigation. Echolocation can shape nearby space with astonishing precision, yet its reach is local. It can define branches, cave walls, insects, and water surfaces close ahead. For journeys stretching across entire landscapes, bats require another guide, one written through the planet itself. Some species appear to sense Earth's magnetic field. This ability, called magnetoreception, gives an animal directional information beyond sight and sound. In bats, it does not replace other senses. It works alongside them, supplying a broad compass while vision, scent, memory, and echoes refine the final approach. Experimental evidence comes from controlled displacement studies with big brown bats. Researchers exposed captured individuals to artificially rotated magnetic fields before releasing them away from their roosts. When the bats departed, their flight directions shifted in ways consistent with the altered magnetic field. They were not merely reacting to landmarks or drifting by chance. Their bodies had read the false compass and responded accordingly. The physical basis may involve magnetite, a naturally magnetic iron oxide mineral found in tissues of several animals. In bats, candidate particles have been reported near the face and neck regions. Magnetite crystals can respond to magnetic fields by rotating or exerting minute mechanical forces. Within sensitive cells, those tiny movements may be converted into nerve signals, giving the brain information about direction. At close range, echolocation remains unmatched. Many bat calls function effectively across only about 5 to 15 meters, which is about 16 to 49 feet. That range is perfect for avoiding a branch or tracking a moth. It becomes almost meaningless when a bat must cross hundreds of kilometers between seasonal roosts. Some migratory species travel more than 1,000 kilometers, which is about 621 miles, between summer and winter habitats. They pass over forests, farms, rivers, ridges, cities, and weather systems, sometimes across routes they have flown only once before. Months later, they may return to a cave entrance or roosting structure with a precision that cannot be explained by echo alone. Brazilian free-tailed bats show how powerful this broader navigation can become. Radar tracking has recorded individuals flying at altitudes exceeding 3,000 meters, which is about 1.9 miles above sea level. With favorable winds, their ground speeds have surpassed 160 kilometers per hour, which is about 99 miles per hour. At that pace, a bat becomes more than a nocturnal hunter. It becomes a long-distance aerial traveler moving through continental scale space. The navigation system appears layered. Magnetic sensing may establish general heading. Vision can recognize larger landscape patterns where light allows. Smell may help identify familiar roost environments. Echolocation then takes over near the destination, resolving the details that matter in the final meters. Such integration makes bat navigation one of the most sophisticated systems known in flying animals. It is neither a single sense nor a single map. It is a partnership among signals, each operating at a different scale. Across darkness, altitude, wind, and unfamiliar terrain, the bat reads the near world in echoes and the far world through Earth's magnetic field, carrying itself home by senses humans can only study from the outside. Some evolutionary partnerships become so precise that each participant seems shaped by the outline of the other. In a cloud forest of Ecuador, one such relationship reaches an almost improbable degree of refinement, linking a bat and a flower through structures that match with extraordinary accuracy. The tube-lipped nectar bat, Anora fistulata, entered scientific literature in 2005. When researchers measured its feeding apparatus, they encountered a record unlike any known among mammals. The bat's tongue could extend to 8.5 centimeters, a distance equal to roughly 150% of its body length. No other mammal possesses a tongue so long relative to the size of the animal carrying it. The arrangement creates an anatomical puzzle. When not in use, the tongue cannot fit entirely inside the mouth. It folds backward through the throat and extends into the chest cavity, occupying space between the sternum and the heart. Muscles, connective tissues, and blood vessels coordinate a structure that can remain compact during flight and then unfurl in an instant when nectar becomes available. Its primary floral partner presents an equally specialized design. The plant, Centropogon nigricans, produces blossoms with nectar chambers buried deep within tubes measuring roughly 8 to 9 centimeters. For most pollinators, the reward is unreachable. Hummingbirds cannot reach the nectar. Insects cannot reach it. Other bats fall short. The tube-lipped nectar bat alone carries the necessary tool. Each feeding visit transfers pollen from flower to flower. Through countless generations, floral depth and tongue length appear to have influenced one another. A slightly deeper flower favored bats capable of reaching farther. A slightly longer tongue favored flowers positioned beyond the reach of competitors. Selection acted on both sides of the partnership, gradually tightening the fit until each became closely linked to the continued success of the other. This relationship represents an extreme expression of a broader pattern. Across the Americas, hundreds of flowering plant species depend on bats for pollination. Different bat lineages evolved tongues, snouts, feeding behaviors, and sensory adaptations suited to particular floral architectures. Plants responded in turn, adjusting bloom shape, nectar production, scent, timing, and position. The result is a network of biological partnerships refined over tens of millions of years. In the forests of Borneo, another plant followed a different path. The pitcher plant Nepenthes hemsleyiana forms elongated chambers used by Hardwick's woolly bat as daytime shelters. Researchers discovered that the pitcher possesses surfaces reflecting echolocation calls with unusual efficiency. To a passing bat, the plant functions almost like an acoustic beacon, producing a distinctive echo pattern that is easy to recognize. Once the bat settles inside, the partnership continues. Waste deposited into the pitcher supplies nutrients that the plant absorbs. In some populations, bat-derived material contributes a substantial portion of the plant's nitrogen intake. A shelter becomes a feeding strategy, and a feeding strategy becomes a shelter. Far to the north, agave plants open their flowers after sunset and present nectar at heights and angles suited to visiting bats. Lesser long-nosed bats carry pollen between blooms across desert landscapes, supporting the reproduction of plants that underpin major agricultural industries today. Taken together, these relationships reveal coevolution in one of its clearest forms. Over immense spans of time, bats and plants have shaped one another so thoroughly that many now function as living components of the same biological design. A bat hanging motionless from a cave ceiling can appear suspended between activity and stillness, occupying a physiological state that stretches ordinary definitions of life. Within that small body, systems that normally operate at a relentless pace begin to slow until their activity becomes almost imperceptible. Many bat species enter daily torpor, a controlled reduction of metabolism that allows survival during periods when food is scarce or environmental conditions become unfavorable. Unlike ordinary sleep, torpor transforms the body from the inside out. Energy consumption falls dramatically. Internal processes contract toward the minimum required to maintain life. The change can be measured in the rhythm of the heart. During active flight, a bat's heart may exceed 1,000 beats each minute. Once torpor deepens, that pace can fall to as few as 18 to 25 beats per minute. Breathing becomes equally sparse. Observers watching from only a short distance may struggle to detect any movement at all. Body temperature declines until it nearly matches the surrounding air, reducing the energetic cost of maintaining warmth. In species that hibernate through winter, this transformation becomes even more extreme. Core temperatures may approach 0 degrees Celsius or 32 degrees Fahrenheit without freezing the animal. Such tolerance depends partly on specialized lipid compositions within cell membranes. These molecular arrangements remain flexible under conditions that would damage the tissues of many other mammals. Human cells exposed to similar circumstances would suffer severe injury, yet bat tissues continue functioning through months of metabolic suppression. Deep hibernation pushes the limits further still. Some individuals have been recorded taking a single breath only once every 2 hours. A little brown bat may remain in continuous hibernation for more than 7 months, surviving entirely on fat reserves accumulated before winter arrived. Every heartbeat, every breath, and every cellular process operates under strict energetic economy. Researchers increasingly view torpor as something more profound than extended sleep. Biological activity is reduced across entire organ systems, approaching a state of temporary suspension. Even more striking is the ability to repeat the process year after year without measurable deterioration of major organs. The heart resumes normal function. The brain resumes normal function. The tissues return to activity with a reliability that remains difficult to explain fully. Periodic awakenings occur throughout hibernation. During these intervals, metabolic activity surges upward before dropping once more. Evidence suggests that these brief returns to wakefulness may allow memory processing, cellular maintenance, and removal of accumulated metabolic waste products. The transition itself appears to serve important biological functions that cannot occur during the deepest phases of torpor. The timing of these arousals adds another layer of mystery. Studies indicate that awakening episodes do not consistently track external temperature changes. Many bats seem to follow an internal schedule governed by biological mechanisms that remain only partly understood. Somewhere within the chemistry of their cells, a clock continues operating, even while much of the body approaches dormancy. Medical researchers study these abilities with growing interest. Bat tissues tolerate prolonged reductions in oxygen availability that would rapidly damage human organs. Understanding those mechanisms could improve organ preservation, transplantation, trauma care, stroke treatment, and cardiac surgery. For medicine, torpor represents a frontier still largely unexplored. For the bat, it is simply another adaptation woven into daily existence, a reversible pause in the machinery of life, refined across millions of years and resumed whenever conditions demand it. The first sign appeared on cave walls, on wings, and across small sleeping faces. During the winter of 2006 to 2007, a photographer documenting bats in Howe's Cave, New York, saw bodies where living clusters should have been. Some bats clung weakly to stone. Others lay dead below the roost. A pale powder marked their muzzles and wing membranes. Nothing in North American bat surveys had prepared researchers for what followed. The condition became known as white nose syndrome, and its cause was eventually identified as Pseudogymnoascus destructans, a cold-tolerant fungus believed to have arrived from Europe. European bats had shared landscapes with it long enough to develop tolerance. North American bats had not. The fungus found its opening in hibernation. During winter, many bats lower their metabolism so deeply that their defenses become limited. They cannot groom normally. Their immune responses are reduced. Their wings, exposed and folded against the body, become surfaces where the fungus can grow. It penetrates delicate membrane tissue, disrupts water balance, irritates nerves, and disturbs the fragile physiology that keeps torpor stable. The most lethal damage comes from forced awakening. A hibernating bat survives winter by rationing fat reserves accumulated before insects disappear. Each arousal burns precious energy. When the fungus wakes bats too often, they exhaust those reserves long before spring. Many emerge into freezing air, searching for insects that are not there. The infection opens the door, yet starvation often delivers the final blow. The spread moved with devastating speed. By 2012, white nose syndrome had reached 19 United States states and 4 Canadian provinces. By 2023, it had been confirmed across 40 states and 7 provinces. Cave by cave, winter survey numbers collapsed. The little brown bat suffered among the heaviest losses. Once one of the most common bats in eastern North America, it declined by more than 90% at many affected hibernation sites. Places that had held tens of thousands, hundreds of thousands, or even millions of wintering bats were reduced to scattered survivors. Some survey teams returned to familiar cave systems and found only a fraction of the life recorded in earlier decades. The Indiana bat entered the disaster already endangered. Since the arrival of the syndrome, it has lost an estimated 80% of its remaining population in many affected regions. Other species, including northern long-eared bats and tricolored bats, followed similar downward paths. The scale of mortality turned a fungal disease into one of the fastest wildlife collapses ever documented among North American mammals. Its speed surpassed many better-known conservation tragedies because it unfolded underground, beyond ordinary view, no open plain filled with bones. No shoreline carried the evidence to passing eyes. The collapse advanced through caves, mines, and winter roosts in places most people would never enter. Even now, the fungus continues to spread into new regions. Some colonies have stabilized at drastically reduced numbers, others remain vulnerable each winter. White nose syndrome revealed how quickly a specialized life can unravel when an introduced pathogen finds the precise weakness in its biology, and how much of an ecosystem can vanish before the surface world notices. Deep within caves that once held immense winter colonies, a small fraction remained. After years of collapse, the numbers stopped falling toward extinction and settled at a fragile level. In some of the hardest hit hibernation sites, surviving populations stabilized at roughly 1 to 5% of their former size. The losses remained staggering, yet those remaining animals carried a message hidden within their biology. Conservation biologists soon realized that survival was unlikely to be a matter of chance alone. If millions had died while a few endured repeated exposure to the same pathogen, some difference must exist between the two groups. That possibility transformed the survivors from the final remnants of a catastrophe into the focus of one of the most important investigations in modern wildlife science. Genetic studies began searching for clues. A study published in 2019 identified variants within surviving little brown bat populations linked to stronger local immune responses in wing membrane tissue. Other genetic differences appeared connected to metabolic regulation during torpor. Together, these traits may allow certain bats to maintain a limited defensive response even while their bodies operate in a reduced physiological state. Such protection would not eliminate infection, yet it could shift the balance enough to survive a winter that proved fatal for others. Changes have appeared in behavior as well. Researchers monitoring surviving colonies observed hibernation patterns that differ from those recorded before white nose syndrome emerged. Some bats now spend shorter, uninterrupted periods in torpor, waking more frequently, and reducing the continuous window available for fungal growth. This strategy carries a cost because every awakening consumes stored energy. Survival depends on entering winter with sufficient fat reserves, creating a delicate trade between resisting the fungus and preserving fuel until spring. Evidence from across the Atlantic Ocean offers another piece of the puzzle. European bats share caves with the same fungus throughout much of their range. They often carry the pathogen, yet widespread mortality rarely follows. Generations of exposure have allowed natural selection to shape populations that tolerate the infection. The fungus remains present, but the relationship has become less destructive. That comparison suggests resistance is biologically achievable. The difference separating vulnerable and tolerant populations is not an impossible barrier. It is an evolutionary path that has already been traveled elsewhere. Researchers at institutions, including the University of California, Santa Cruz, and several Canadian organizations, are examining whether genetic markers associated with fungal tolerance can be identified and incorporated into conservation planning. Other efforts focus on assisting bats directly. Beneficial microorganisms capable of competing with the fungus are being tested as probiotic treatments in multiple cave systems. Early trials have shown measurable reductions in fungal growth on treated animals and surfaces. Laboratory experiments have also revealed that carefully controlled exposure to a specific wavelength of ultraviolet light can kill the fungus on bat skin without producing measurable harm to the bats themselves. Field systems designed to deliver this treatment are now under evaluation. Each survivor, therefore, represents more than a single animal. Within its cells may exist combinations of genes, physiological responses, and behavioral adaptations refined by intense selection. Those living bats form a library written by survival itself. Learning how to read that library may help restore populations that once filled the darkness with millions of wings, and may also deepen our understanding of how living systems adapt when confronted by a new and relentless threat. Across tropical forests, deserts, farms, and grasslands, countless ecological relationships continue each night without announcement. A flower opens after sunset. A fruit ripens on a distant branch. A cloud of insects rises above a field. In each case, bats enter a chain of events that extends far beyond their own survival, linking their movements to forests, food systems, and economies spread across continents. More than 500 plant species, representing at least 67 plant families, depend on bats for pollination. This places bats among the most influential pollinators on Earth, alongside bees and butterflies. Their contribution is often less visible because much of it occurs after dark, when flowers adapted for bat visitors release nectar and scent into the night air. One of the clearest examples grows in the dry landscapes of Mexico and the southwestern United States. Wild agave plants rely heavily on the lesser, long-nosed bat for pollination. The beverages tequila and mezcal originate from agave, supporting an industry worth many billions of dollars each year. Commercial cultivation frequently reproduces plants through cloning, reducing immediate dependence on pollinators. Yet, wild populations preserve the genetic diversity that allows adaptation to disease, drought, and environmental change. Without bat pollination, that reservoir gradually shrinks. Far across Southeast Asia, another economic story unfolds. Durian, among the region's most valuable fruit crops, depends largely on cave nectar bats. These bats transfer pollen between flowers during feeding visits, allowing fruit production on a scale that supports growers, traders, transport networks, and regional markets. The connection is direct. Remove the pollinator, and fruit production falls with it. Certain partnerships extend far beyond commerce. The baobab tree, an iconic species across sub-Saharan Africa, produces flowers adapted to bat visitation. Some individual trees have persisted for more than 2,000 years. Their continued reproduction depends largely on flying mammals carrying pollen between blooms. A relationship measured in millennia continues through nightly encounters lasting only moments. Fruit bats contribute in another way. Seeds consumed in one location may be deposited many kilometers away. Recorded dispersal distances can exceed 57 kilometers, which is about 35.4 miles, within a single night. Few other vertebrates transport seeds so effectively across fragmented landscapes. In regions shaped by logging, agriculture, or fire, these journeys help reconnect isolated patches of vegetation. New forests often begin with seeds carried by animals capable of crossing gaps that ground dwellers cannot. The influence of bats also reaches agricultural pest control. A colony of 150 big brown bats can consume approximately 1.3 million pest insects in a year. Across North America, estimates of the economic value provided by bat insect suppression range from several billion to more than $50 billion annually. The exact figure remains debated, yet every calculation points toward the same conclusion. Bats remove enormous numbers of crop-damaging insects. When local bat populations decline, pesticide use frequently rises. Farmers compensate for the disappearance of natural pest control with chemical alternatives. A landmark study, published in 2011, demonstrated that bat losses linked to white nose syndrome carried major agricultural costs, revealing one of the largest ecological services operating largely without public recognition. Forests expanded, crops flourished, and agricultural systems developed, while bats continued feeding, pollinating, dispersing seeds, and consuming insects. Over tens of millions of years, human economies and natural ecosystems became intertwined with those activities so completely that the connection often faded from view. The world that surrounds us bears the imprint of wings moving through darkness. A colony of bats can sound chaotic to human ears. Thousands of calls overlap, individuals shift positions, brief disputes erupt and fade. Mothers search for offspring, and neighbors negotiate personal space. For a long time, natural history treated this activity as background noise surrounding otherwise solitary animals. Careful observation revealed something very different. Inside many bat colonies exists a social world structured by familiarity, memory, communication, and long-term relationships. Greater spear-nosed bats provide one of the clearest examples. Females form stable social groups that persist across years. The same individuals repeatedly choose to roost together, travel through overlapping foraging areas, and respond collectively when danger approaches. Field observations have documented coordinated mobbing behavior directed toward predators, with several bats joining defensive responses that protect members of their group. Such cooperation depends upon recognition. An individual must distinguish companions from strangers and maintain those distinctions across extended periods. Communication helps maintain these relationships. Egyptian fruit bats have become the focus of one of the most detailed investigations ever conducted into bat vocal behavior. Researchers at Tel Aviv University recorded tens of thousands of vocalizations from colony members and analyzed them using advanced computational methods. Patterns emerged from what initially appeared to be an indecipherable stream of sounds. More than 60% of recorded vocalizations were directed toward specific individuals rather than broadcast generally throughout the colony. The information encoded within those exchanges proved equally striking. Analytical models could identify the caller, the individual receiving the call, and the context surrounding the interaction. Some vocalizations occurred during feeding disputes. Others accompanied mating attempts, competition for roosting positions, or unwanted physical contact. Distinct acoustic structures corresponded to different social situations. The calls functioned less like simple signals and more like targeted social messages exchanged between recognizable individuals. These findings challenged assumptions that had long separated bat communication from the communication systems studied in primates. Directed address, individual identity recognition, and context dependent vocal variation all satisfy criteria previously associated with much larger brains and more familiar social mammals. The colony began to appear less like a crowd and more like a network of ongoing relationships. Geography adds another layer. Young bats, raised in separate regions, often develop vocal patterns that differ measurably from those of neighboring populations. Researchers describe these differences as vocal dialects. The underlying sounds remain understandable within the species, yet local populations acquire distinctive characteristics shaped by social learning. Similar processes contribute to regional accents in human speech, where communication remains possible despite recognizable variation. Early life offers another glimpse into the flexibility of bat communication. During the weeks following birth, pups gradually adjust their calls in response to maternal feedback. Their vocalizations drift toward the characteristic patterns used by their mothers. The process resembles vocal learning observed in human infants, where repeated interaction guides the development of increasingly refined sounds. Social attachment extends beyond communication alone. Experimental studies have documented measurable physiological consequences when long-term companions are separated. Stress hormone levels rise, resting behavior changes, and foraging efficiency declines. Reunion reverses many of these effects, suggesting that established social bonds influence daily functioning in meaningful ways. The familiar image of a lone bat hanging apart from others survives largely because so much of bat life unfolds beyond ordinary human perception. Within caves, forests, and roosts around the world, conversations continue, alliances persist, and relationships endure. Darkness conceals the details, yet the social lives unfolding there often resemble patterns found throughout the broader mammalian world. Human beings met bats mostly at the edges of perception. They appeared at dusk, moved too quickly to study easily, vanished into caves, and returned from places associated with depth, night, and transformation. Across continents, this partial visibility gave them unusual symbolic power. Cultures with no contact with one another built stories around the same animal, and those stories moved in strikingly different directions. In Mesoamerica, the Maya world gave the bat one of its most dramatic forms. Camazotz appears in the Popol Vuh as a death bat associated with the underworld, sacrifice, night, and transition. The figure belonged to a landscape where caves carried spiritual importance and where real bats occupied spaces used for shelter, ritual, and burial. Archaeological bat imagery in Mesoamerican cultures dates back to at least 300 years before the Common Era, showing that this association reached deep into regional memory. Some researchers have suggested that Camazotz may have drawn inspiration from Desmodus draculae, a giant vampire bat that survived until roughly 10,000 years ago. Its wingspan approached 50 centimeters, which is about 19.7 inches. Early people living near caves in the region may have encountered remains, living populations, or inherited stories of a larger blood feeding bat, blending animal reality with sacred imagination. In China, the symbolic direction changed completely. The Mandarin word for bat, bianfu, resembles the word for good fortune through sound association, and bats became signs of prosperity, health, longevity, and happiness. Across more than 2,000 years of art, ceramics, clothing, furniture, and architecture, bat designs appeared as blessings rather than omens. The five blessings motif arranged five bats in a circular pattern representing long life, wealth, health, virtue, and a peaceful death. In ancient Egypt, bat imagery appeared in protective contexts connected to passage through the afterlife. An animal able to move through darkness without apparent confusion became an emblem of guidance through unknown space. The bat's body, with its folded wings and nocturnal habits, offered a natural symbol for transition between worlds. Indigenous Australian traditions also preserved bat stories, often tied to ecological knowledge. In some regions, bat activity helped mark seasonal insect emergence, weather shifts, and the location of important roost trees. Such stories were never only decorative. They carried information across generations, binding observation to memory through narrative. Europe developed a darker inheritance. Medieval Christian art often associated night creatures with danger, disorder, and moral uncertainty. Because bats emerged between day and night, they became linked with boundaries. Witches flew. Devils gained wings shaped like bat membranes. Later, vampire folklore absorbed the biology of Latin American blood, feeding bats after European descriptions circulated in the 16th century. Bram Stoker's Dracula, published in 1897, fused those traditions into an enduring modern image. The same animal became death god, blessing, guide, weather sign, demon wing, and vampire companion. Such variety reveals less about bats alone than about the human imagination, meeting a creature difficult to see, difficult to hear, and impossible to place neatly within ordinary categories. The bat lived at the edge of the known world, and culture followed it there. Many of the technologies woven into modern life began with a simple biological question: how does a bat move through darkness without colliding with branches, cave walls, or one another? The search for that answer opened a chain of discoveries that eventually reached hospitals, aircraft laboratories, renewable energy research centers, and advanced robotics facilities. Few animals have contributed so much to human technology while asking nothing in return. The story began with skepticism. In 1938, zoologist Donald Griffin and physicist Robert Galambos presented experimental evidence showing that bats navigate using high-frequency sound and returning echoes. The conclusion challenged assumptions held by much of the scientific community. One editor reportedly dismissed the idea as sheer fantasy. The proposal seemed too strange to fit existing understanding. Yet repeated experiments produced the same result. Bats were constructing acoustic maps of their surroundings through sound. Griffin introduced the term echolocation, and the principle soon attracted attention far beyond zoology. During the Second World War, researchers developing sonar systems found themselves working with the same physical concepts already demonstrated in bats. Sound pulses moved outward, reflected from objects, and returned with information about distance and location. Engineers transformed that principle into tools that helped ships detect hazards and submarines beneath the ocean surface. Nature had provided a functioning prototype tens of millions of years earlier. The same foundation eventually entered medicine. Modern ultrasound imaging relies upon emitted sound waves and returning echoes to reconstruct structures hidden within the body. Hospitals across the world use this technology every day, whether examining developing infants, monitoring organs, or diagnosing disease. The process follows the same fundamental logic employed by a hunting bat navigating through darkness. Researchers also looked more closely at the remarkable hearing system that makes echolocation possible. The inner ear of a bat can distinguish tiny differences in frequency and timing. Studies of this architecture have informed cochlear implant development, particularly in signal processing strategies designed to improve how artificial hearing systems interpret sound. Bat hearing became a biological benchmark for engineers attempting to restore human hearing. Attention then shifted toward the wing itself. The patagium, with its elastin and collagen network, combines flexibility, durability, and self repair in ways that synthetic materials still struggle to reproduce. Scientists studying soft robotics, prosthetic skin, and flexible electronics continue to examine how these membranes stretch, recover, and heal after damage. The engineering challenge remains unfinished, but the biological model is already available. Flight researchers found inspiration there as well. Scalloped leading edges, observed in bat wings and certain whale fins, have been applied to experimental wind turbine blades. Prototype testing has demonstrated roughly 32% improvements in energy capture under some low wind conditions compared with conventional, smooth edge designs. Robotics laboratories have pushed even further. The BatBot, developed through collaboration between the California Institute of Technology and the University of Illinois, uses flexible membrane wings, articulated joints, and embedded sensors to mimic bat flight. Its performance in cluttered indoor spaces highlights maneuverability that many conventional drones still cannot match. Even vampire bats have contributed. Desmoteplase, derived from anticoagulant compounds found in vampire bat saliva, has been investigated as a treatment for ischemic stroke. Researchers continue evaluating its therapeutic potential and clinical applications. The bat never set out to advance medicine, robotics, aviation, or engineering. It was solving the problems of flight, navigation, feeding, and survival. Human technology continues to discover how sophisticated those solutions truly are. A forest can appear unchanged from a distance. Trees still stand, leaves still move in the wind, and the familiar outline of the landscape remains. Yet within that landscape, relationships can begin to unravel long before the damage becomes obvious. The story of bats and emerging diseases begins in that hidden layer of ecology, where the health of an ecosystem and the health of human communities are tied together more closely than they first appear. For many people, learning that bats can carry viruses such as Ebola, severe acute respiratory syndrome, Marburg virus disease, and Nipah virus leads to an immediate conclusion. Fewer bats might seem safer. Research conducted across multiple continents has steadily pointed toward a different reality. The greatest risks often emerge where bat populations are stressed, displaced, or forced into closer contact with humans through environmental disruption. Evidence from Nipah virus outbreaks in Bangladesh and Malaysia provides one of the clearest examples. These outbreaks, which have recorded fatality rates ranging from roughly 40% to 75%, repeatedly occurred in landscapes where forests had been fragmented or reduced. As natural habitat disappeared, bat colonies shifted into agricultural areas, villages, and regions occupied by livestock. encounters that had once been uncommon became increasingly frequent. Scientists studying these colonies found important differences between stable populations and disrupted ones. Bats living in intact habitat generally maintained predictable movement patterns and consistent viral dynamics. Colonies experiencing food shortages, habitat loss, or repeated disturbance showed broader ranging behavior and elevated viral shedding. Saliva, urine, and feces contained higher concentrations of viruses that normally remain present at low levels without causing illness in the bats themselves. The mechanism appears to involve physiological stress. Nutritional strain and habitat instability affect immune regulation, altering the balance that has developed between bats and the viruses they host. Under those conditions, viral replication can increase. The pathogens are not created by environmental destruction, they already exist within the ecological system. Habitat disruption changes the circumstances under which exposure becomes more likely. This understanding has transformed how many disease ecologists think about prevention. Removing forests does not remove viruses. Eliminating roosts does not eliminate reservoirs. Actions intended to separate people from wildlife can sometimes achieve the opposite effect by pushing animals into human dominated landscapes. The forest itself functions as a buffer, maintaining distance, stability, and ecological structure. Analyses published in the journal One Health during 2020 found that forest cover loss predicted Nipah spillover risk more accurately than bat population size across parts of South and Southeast Asia. Similar patterns have been documented in studies of Ebola virus emergence in Central and West Africa. Spillover events frequently cluster near recently disturbed habitats where human activity intersects with displaced wildlife populations. North America presents a different situation. The collapse of bat populations caused by white nose syndrome has not produced measurable increases in bat associated viral spillover, according to current evidence. Researchers nevertheless continue monitoring surviving colonies as distributions shift and new roosting patterns emerge. Ecological systems rarely remain static after major population declines. Viewed through this lens, bat conservation and pandemic prevention cease to be competing goals. They become parts of the same strategy. Healthy forests support stable bat colonies. Stable colonies reduce opportunities for dangerous contact. The bat is not a threat standing outside the system. It is one thread within a larger fabric, and the strength of that fabric depends on keeping the whole pattern intact. Inside every bat cell is a record older than any fossil yet found. Bone can vanish, membranes can decay, and whole chapters of ancestry can pass without entering stone. The genome keeps a different archive. It preserves traces of pressure, survival, and change in the language of deoxyribonucleic acid, written across millions of years in patterns researchers are only beginning to read. In 2020, a major study published in Cell compared the genomes of 40 bat species. At the time, it was among the most comprehensive genomic surveys ever conducted on the order. The results revealed more than 1,000 genomic regions showing accelerated positive selection compared with equivalent regions in other mammals. Evolution had acted on bat deoxyribonucleic acid with unusual speed and consistency. These changes did not gather around one trait. They appeared across immune systems, metabolic pathways, sensory genes, developmental controls, and repair mechanisms for damaged genetic material. The pattern suggested a body being reshaped across multiple systems at once: flight, hearing, longevity, Viral tolerance and cellular maintenance were not separate stories stacked beside one another. They were connected through the same biological transformation. The immune changes fit what researchers already saw in living bats. Modifications in the stimulator of interferon genes, pathway, and interferon regulation help explain how bats tolerate viruses while limiting destructive inflammation. Their defenses remain alert without constantly damaging the body they protect. The genome shows that this balance was built through deep evolutionary adjustment. Other regions point toward long life. Genes involved in repairing damaged genetic material and maintaining cellular stability show signs of selection in several bat lineages. These changes connect with the unusual lifespans already observed in small bats that live far longer than body size predicts. The genome does not merely confirm the longevity paradox, it begins to explain how that paradox became possible. Sensory genes tell another part of the story. Changes associated with hearing, including the gene known as Preston, reveal selection on the machinery of sound perception. The cochlea, auditory pathways, and neural timing systems required for echolocation were supported by molecular shifts that sharpened the bat's relationship with reflected sound. Metabolism forms the central thread, tying much of this together. Powered flight demands enormous energy. High energy demand can produce cellular stress and damage. Selection favored more efficient energy systems, stronger repair pathways, and dampened inflammatory reactions. Those same adaptations helped support longevity and viral tolerance. One pressure created many consequences. This is the deeper meaning of the bat genome. Evolution did not merely add one strange feature after another, it altered a network. Each adaptation made others more viable. A wing changed metabolism. Metabolism shaped immunity. Immunity influenced viral tolerance. Repair systems supported long life. Sensory changes opened the night. Traditional explanations often describe adaptation as individual traits selected one at a time. Bats reveal a more integrated possibility. Under intense pressure, with a flexible developmental toolkit and access to new ecological space, evolution can tune several linked systems together. The bat is therefore not a scattered collection of biological oddities. It is a coherent argument written in living tissue, refined across more than 50 million years, and still unfolding each time a genome is read. A bat hanging from a cave ceiling can seem less like a familiar mammal and more like a challenge. Every feature we have followed through this journey, flight, echolocation, viral tolerance, longevity, torpor, and social complexity, gathers into a single question about the nature of change itself. The animal becomes a lens through which evolution can be viewed at a different scale. Popular imagination often pictures evolution as a procession of tiny steps. Generation follows generation, each nearly identical to the last, while change accumulates so gradually that it becomes visible only across immense stretches of time. That image contains truth, yet the history of bats suggests that it is only part of the story. When the earliest known bats appear in the fossil record, they arrive carrying much of the anatomy that defines them today. Wings are already present. Flight is already established. The skeletal framework required for aerial life is already functioning. Soon afterward, bats diversify into an expanding array of ecological roles. Paleontologists, searching for long sequences of intermediate forms, have found far fewer than expected. Their genetic history points in the same direction. Comparative genomic studies reveal accelerated positive selection across more than 1,000 regions of the bat genome. Those changes do not belong to a single biological system. They appear in genes associated with immunity, sensory processing, metabolism, development, and cellular maintenance. Evolution was acting across an interconnected network rather than refining one isolated trait at a time. Evolutionary developmental biology provides a useful framework for understanding this pattern. Vertebrates share a deeply conserved genetic toolkit. Limb formation, body patterning, organ development, and growth regulation rely on systems inherited from distant ancestors. The crucial changes often occur within regulatory sequences that control when, where, and how strongly genes are expressed. From that perspective, a bat wing is not an entirely new invention. It is a mammalian hand whose developmental instructions have been adjusted. Finger bones lengthen dramatically, membranes expand between them, growth patterns shift, and a structure once adapted for climbing and grasping becomes capable of powered flight. The same principle appears elsewhere. Echolocation builds upon auditory machinery already present in ancestral mammals. Immune adaptations modify existing defense pathways. Longevity emerges through alterations to mechanisms governing repair, inflammation, and cellular stability. Novelty arises through reorganization of inherited components. This means that the potential for transformation often exists long before transformation occurs. Biological systems contain possibilities that remain unrealized until ecological conditions, selection pressures, and developmental flexibility align. The raw materials do not need to be created from nothing. They already reside within the organism. The fossil pattern of bats aligns closely with the concept of punctuated equilibrium proposed by Stephen Jay Gould and Niles Eldredge. According to this view, long intervals of relative stability can be interrupted by comparatively rapid bursts of evolutionary change. The pace is not constant. History can move unevenly. Bats illustrate that evolution has no fixed speed limit. Under sustained directional selection, supported by developmental systems capable of responding, change can proceed with surprising intensity. What appears impossible when viewed as a finished product becomes understandable when examined as a process. The bat does not stand outside evolutionary theory as an exception demanding special treatment. It stands within it as a demonstration of its full capacity. In that sense, the animal reveals something larger than itself. Evolution is not merely gradual modification. It is a dynamic exploration of possibilities already woven into life. A raindrop striking a pond, the edge of a leaf trembling in moving air, the thin line of a branch extending across open space. To human awareness, these details belong mostly to sight. For a bat, they belong to sound. The same physical world exists around both animals, yet each encounters it through a profoundly different doorway. Biologist Jakob von Uexküll introduced the concept of the Umwelt in 1909, describing the species' specific sensory universe inhabited by every living creature. No animal experiences reality in its entirety. Each receives a limited stream of information filtered through the capabilities of its body. From those signals, the brain constructs a working model of existence. Human beings live largely within a visual umwelt. Color, contrast, texture, movement, and distance arrive through light, reflected from surfaces and focused onto the retina. Neural circuits transform those signals into a coherent spatial landscape. The experience feels complete because it is all we have ever known, yet it represents only a narrow fraction of the information available in the surrounding environment. The bat occupies a different sensory domain. During active hunting, many species emit echolocation calls at rates exceeding 200 pulses per second. Each pulse expands outward through space, strikes objects, and returns as a stream of echoes. Those returning signals carry information about distance, direction, movement, texture, and shape. The bat's brain assembles this information into a continuously updated, three-dimensional representation of its surroundings. Within that acoustic landscape, distance emerges from echo delay. Velocity reveals itself through changes in frequency caused by motion. Surface texture appears through subtle alterations in returning sound patterns. The flutter of an insect wing produces a recognizable signature. The contour of a cave wall, the angle of a wire, and the ripples crossing a pond all become detectable features. Information arrives so rapidly that the animal can adjust its flight path in fractions of a second. It can be tempting to imagine this as a diminished version of human perception, a substitute developed for life away from daylight. The evidence points elsewhere. Echolocation grants access to properties of the physical world that human senses cannot directly detect. The bat is not compensating for a missing experience. It is participating in a different one. This realization inspired philosopher Thomas Nagel's influential essay, published in 1974, titled, "What Is It Like to Be a Bat?" His argument centered on consciousness itself. Even if scientists mapped every neural pathway and decoded every echolocation signal, the subjective experience produced by that biology would remain inaccessible to a human mind built upon different sensory foundations. Nagel's point was not that bats possess mystical abilities, it was that experience depends upon embodiment. A nervous system shapes the reality available to its owner. Different bodies, therefore, generate different experiential worlds, each valid within its own frame of perception. When a bat releases an echolocation pulse and receives the returning echoes, it is not navigating a version of the scene a human observer would recognize. It is moving through an acoustic reality constructed from information our senses never evolved to collect. The night, from that perspective, becomes richly structured and continuously revealed. A creature sharing our skies for roughly 50 million years has inhabited that alternative sensory world all along, carrying with it a reminder that reality is always larger than any single way of perceiving it. A roof beam, a barn rafter, the underside of a bridge, the sheltered lip of an old building. These are ordinary human structures, yet for bats, they can become cliff faces, cave mouths, and nursery walls. Wherever people have built places with shade, height, and narrow openings, bats have found ways to live near us without fully entering our attention. They have shared human environments for thousands of years. Across inhabited continents, they roost in roof spaces, church towers, abandoned mines, bridge joints, tree cavities beside farms, and caves near settlements. Their presence is often discovered through small signs rather than direct encounters. A flutter at dusk, droppings below a beam, faint movement behind wood, the brief crossing of wings against the last light. In Austin, Texas, this overlap becomes visible on a grand scale. The Congress Avenue Bridge hosts roughly 1.5 million Mexican free-tailed bats, the largest urban bat colony known in the world. Beneath a bridge crossed each day by commuters, tourists, cyclists, and schoolchildren, a mammalian city waits for evening. When the colony emerges, the sky fills with bodies that had been resting all day below traffic and concrete. Their invisibility has little to do with rarity. It comes from timing. Bats occupy the hours that human routines often abandon, the period after sunset, the deep night, and the edge of dawn. They move through the same streets, gardens, rivers, orchards, parks, and forest edges that people use by day, shifted into another schedule. The separation is temporal as much as spatial. During those hours, the animal above a roofline may be doing almost everything this story has traced. It may be reading nearby space through returning sound. It may be following a route remembered from past nights. It may be carrying pollen between flowers, dispersing seeds beyond a forest gap, removing insects from a field, or returning to a roost where social bonds, offspring, and familiar voices wait. Some individuals travel immense seasonal distances. A migratory bat crossing 1,000 kilometers, which is about 621 miles, carries within its body a magnetic sense, memory of landscape, and the aerodynamic inheritance of more than 50 million years. Others remain close to a single neighborhood, hunting above ponds and street trees, while generations of people pass below without noticing. Human landscapes are partly bat-shaped even when no bat is visible. Forest edges regrow from seeds carried in flight. Agave, durian, baobab, and countless other plants maintain older partnerships. Agricultural fields benefit from the disappearance of insects that would otherwise feed on crops. The work is ordinary for the bat, yet it leaves consequences that stretch far beyond each night's movement. The history of life is filled with organisms that changed the world without drawing attention to themselves. Bats belong among the most successful of them. Their order has persisted through climate shifts, continental changes, new predators, new diseases, and the arrival of human cities. Still, they remain overhead, entering familiar spaces by routes most people never learn to see. A small shape crossing above a house at dusk contains more than movement. It carries an evolutionary experiment in flight, sound, immunity, memory, and endurance. The bat above your head is not a visitor from the margins of nature. It is part of the structure of the living world, present all along, moving through the same air. The history of biology contains a recurring pattern. An organism appears to violate expectations. Researchers declare a feature improbable or unattainable, and years later, the mechanism emerges from careful study. The boundary between the impossible and the understood has shifted many times, carried forward by discoveries hidden inside living systems. Few animals illustrate that pattern more completely than the bat. When naturalists in the 18th century began examining bat anatomy in detail, flight itself seemed difficult to reconcile with mammalian design. Birds possessed feathers and lightweight frames that appeared suited to the air. Bats flew with skin stretched across elongated fingers, a structure that looked fragile when compared with feathered wings. The arrangement puzzled early observers. Today, the patagium is understood as a sophisticated flight surface containing nerves, blood vessels, muscles, sensory receptors, elastin, and collagen organized with extraordinary precision. What once appeared implausible now informs aerospace research and aerodynamic engineering. A similar story unfolded around lifespan. Comparative biology established one of its most reliable patterns. Small mammals generally live brief lives. Bats refuse to follow that expectation. Species weighing only a few grams survived for decades, outlasting mammals many times their size. The discrepancy appeared so large that it challenged accepted assumptions about aging. Gradually, the underlying biology came into focus. Telomere maintenance systems, altered nutrient sensing pathways, efficient mitochondria, and enhanced cellular repair revealed pieces of the puzzle. The anomaly remained extraordinary, yet it no longer stood outside explanation. Their sensory world produced another chapter in the same story. During the early 20th century, The proposal that bats navigated using high frequency sound struck many scientists as implausible. One editor reportedly dismissed the concept as fantasy. Experiments eventually demonstrated that bats emit calls, receive returning echoes, and construct detailed spatial information from those signals. What seemed impossible became measurable. What seemed mysterious became a field of research. The same physical principles now support technologies used throughout modern medicine. Immune biology offered another surprise. Bats carry viruses that cause severe disease in many other mammals, yet often show little sign of illness themselves. At first glance, the pattern appeared to contradict established ideas about host and pathogen relationships. Subsequent genetic and physiological studies uncovered immune pathways reshaped over millions of years. Modified inflammatory responses, altered signaling systems, and unique antiviral mechanisms helped explain how tolerance could emerge without abandoning defense. Each example follows the same progression. An observation appears inconsistent with prevailing expectations. Investigation replaces disbelief. Mechanisms emerge. Understanding deepens. The animal remains extraordinary, though the sense of impossibility gradually dissolves under evidence. Seen together, these discoveries suggest something broader about evolution itself. The bat is not an exception standing outside biological rules. It is a demonstration of how far those rules can reach when opportunity, selection pressure, and developmental flexibility interact across immense spans of time. Flight, echolocation, longevity, viral tolerance, navigation, and social complexity are woven into a single lineage that continued refining itself for more than 50 million years. Future discoveries will almost certainly uncover other organisms that appear to challenge accepted limits. History suggests a useful response. Apparent impossibility is often an invitation to look more carefully. The bat never violated the rules of life. It revealed how expansive those rules had been all along, waiting to be understood. The bat does not need human admiration to remain what it is. For more than 50 million years, it has crossed the night, found food, raised young, followed seasons, entered caves, and returned to the air without waiting to be understood. Its life began long before human attention existed, and its senses still move through a world our bodies cannot enter. That independence has changed in one important way. In many regions, the future of bats now depends on decisions made by humans. The animal that survived geological upheaval, climate shifts, predators, disease, and ecological change has entered an age where its greatest pressures come from a species that shares its landscapes while rarely sharing its schedule. White nose syndrome offers one example. A fungus moved across continents through human activity and reached North American caves where many bats had no inherited tolerance. Cave exploration, travel, and altered movement patterns helped accelerate a spread far faster than natural dispersal would likely have allowed. Winter roosts that once held great colonies became sites of decline within only a few decades. Habitat destruction presses from another direction. Forest clearing removes roosts, feeding roots, and flowering plants. It pushes colonies towards smaller fragments of suitable habitat, increasing stress and bringing bats closer to farms, towns, livestock, and orchards. The same disruption that weakens bat populations can also increase the chance of viral spillover by breaking the ecological distance that once separated wildlife and people. Other pressures arrive through modern infrastructure. Wind turbines can kill migratory bats during seasonal movements. Pesticides reduce the insects many species depend on for food. Artificial light alters foraging patterns, disrupts travel corridors, and can make traditional roosts unsuitable. Each stressor may appear separate, yet together they form a world increasingly difficult for bats to navigate. The case for protecting them does not require sentiment. Across more than 1,400 species, bats support systems that human life already depends upon. They pollinate plants, disperse seeds, suppress agricultural pests, help maintain forests, and contribute to ecological stability in ways that often remain unnoticed until they disappear. Their presence is part of the living infrastructure beneath food production, forest recovery, and disease prevention. Protection, then, becomes less an act of kindness than an act of maintenance. A bridge cannot lose its supports indefinitely and remain unchanged. A forest cannot lose its seed carriers without consequence. An agricultural region cannot lose natural pest control without replacing it through other means. The systems surrounding human life are full of such hidden dependencies. The bat asks nothing in any human sense. It does not request study, sympathy, mythology, or rescue. Still, the evidence gathered from fossils, genomes, caves, farms, hospitals, and forests has made its own argument. A species capable of decoding bat sonar, sequencing bat genomes, studying bat immunity, and deriving medicine from bat saliva is also capable of noticing the living animal above its head. What the bat asks of us is only attention translated into restraint, less destruction, better habitat, safer energy, fewer careless crossings between wild systems and human systems. It has been present all along, moving through the same air, carrying work older than our species. The question becomes unnecessary when the answer is already visible in the world it helps keep alive. A story can begin with a puzzle and end with a change in perspective. The bat entered this journey carrying a collection of traits that appeared difficult to reconcile with familiar expectations. A mammal had taken to the air. A small body lived far longer than its size seemed to permit. Viruses that devastate other species moved through it with limited consequence. Sound had become a tool for perceiving space. The deeper each mystery was examined, the more each one shifted from impossibility toward explanation. Again and again, the pattern remained the same. Features that once appeared disconnected revealed underlying mechanisms. Flight connected to metabolism, metabolism connected to cellular maintenance, cellular maintenance connected to longevity, longevity intersected with immunity, immunity shaped viral tolerance. Traits that seemed isolated became threads in a single biological fabric woven across millions of years of selection and refinement. That realization changes the meaning of the missing chapter in the fossil record. Earlier generations often viewed the absence of transitional fossils as a problem demanding resolution. Seen through the combined evidence of comparative anatomy, genomics, developmental biology, and living species, the gap becomes something different. It reflects the reality that some transformations unfold in places where preservation is rare, during intervals brief on geological timescales, leaving only fragments for stone to retain. The genome helped illuminate those lost passages. Within living bats, researchers found traces of ancient changes preserved in molecular form. Regulatory systems shifted, immune pathways changed direction. Hearing genes acquired new functions. Developmental programs altered the proportions of the mammalian hand until fingers lengthened into the framework of a wing. The living animal became a record written in biology rather than rock. Across more than 1,400 species distributed through deserts, forests, mountains, islands, cities, caves, wetlands, and grasslands, a larger picture emerges. The bat is neither an exception to evolution nor a challenge to its principles. It is evidence of what those principles can achieve. Given opportunity, flexibility, and sustained selection, life can move into regions of possibility that once seemed inaccessible. The longevity that stretches beyond expected mammalian limits arises from cellular processes that researchers can increasingly trace. The sonar system follows physical laws of acoustics and neural processing. Viral tolerance emerges from modifications within immune regulation. Each discovery narrows the territory occupied by mystery while expanding appreciation for the processes underneath. Nothing supernatural is required. No hidden force waits behind the curtain. Biology itself is sufficient. The mechanisms are real, measurable, and testable. Their complexity can create the impression of improbability, yet complexity and possibility have never been opposites. Even now, while roofs cool after sunset and trees stand against the sky, bats are crossing the air above countless landscapes. They move through spaces shared with human beings, yet experienced through entirely different senses. Their bodies carry histories extending far beyond recorded civilization. Their lives continue according to rhythms established long before human observers began asking questions about them. The darkness overhead remains filled with motion. What has changed is not the bat. The change lies in how that darkness is understood. A place that once seemed empty now contains a lineage shaped by deep time, carrying the record of transformation, adaptation, and persistence. The dark above us is the same dark it has always been. We can finally see a little more of what has been there all along.